DNA replication is pretty efficient, and out of 3 billion base pairs, only a few mistakes are made each time it divides. But DNA can also be damaged by things like ultraviolet light, X-rays, reactive oxygen species, or chemical mutagens. So how bad is a DNA mutation? Well, that depends on what type of mutation it is. I already talked about silent mutations due to the third or wobble base in a codon. Here's the same example as before, where you can see that even if you change one of these third bases, you still get the same amino acid. So this is the least problematic kind because it doesn't really have any effect on the protein. A missense mutation also results from changing one nucleotide, but in this case it'll also change the amino acid sequence that the codon codes for. In this example, we're changing the second base from CCU to CUU, which makes an amino acid change from proline to serine. This can be relatively harmless or extremely harmful, depending on a few things. First of all, if the mutation is in an important part of the protein, such as its active site, even a single amino acid change can prevent some enzymes from functioning. Second, changing from one amino acid to a similar one, such as from one medium-sized nonpolar amino acid to another medium-sized nonpolar amino acid, will usually have a smaller effect than changing from a positively charged to a negatively charged amino acid, or vice versa. Lastly, the severity of the mutation depends on the importance of the protein, since some proteins have more vital functions than others. But generally speaking, missense mutations are usually not that big of a deal. Next is a nonsense mutation, which is when the nucleotide change converts a codon that codes for an amino acid to one that codes for a stop codon. If we look at our codon table, you can see that UUA usually codes for leucine. But if that middle nucleotide is mutated to become an A, then we get UAA, which is a stop codon. Nonsense mutations have a much stronger effect on protein function, since you lose everything downstream from where the mutation occurred, which often has the same effect as deleting the protein entirely. Okay, so everything I talked about so far has been changing one nucleotide base into a different one. A frameshift mutation is unique among all the ones listed here, because it's the only one in which nucleotides are added or removed, rather than just changed. Since this throws off the commonless, non-overlapping code that I mentioned a few minutes ago, everything downstream from the mutation will code for the wrong amino acids. For example, if we have this sequence broken down into these codons by color, we have the sentence, "Can you dig her new hat?" But if we add the letter X right here, all these codons will be shifted over and will now code for something different. So unlike a missense mutation, where you have one different amino acid, now you'll have a lot of different amino acids. Frameshift mutations usually cause a similar amount of damage as nonsense mutations, but in some cases can be dramatically worse. Can you think of why? Nonsense mutations usually just result in a less effective or non-functional protein, whereas frameshift mutations result in a new amino acid sequence which can take on a new function, and these new functions can really throw off cellular homeostasis, potentially leading to cancer or genetic diseases. And in fact, two diseases that are often caused by frameshift mutations are Tay-Sachs and familial hypercholesterolemia. Alright, so let's say we have some of these mutations and we want to fix them. How can we do this? Well, it depends on the type of mutation. Ultraviolet light, which is the part of sunlight that gives you a sunburn, causes adjacent pyrimidine bases in DNA, mostly thymidine, to covalently attach to one another, forming a dimer. This can cause mutations in the next round of DNA replication. This is the most important kind of mutation that is resolved by our first repair mechanism, called nucleotide excision repair. Basically, the enzymes involved in this repair pathway will cut the damaged strand upstream and downstream from the site of the damage, and remove the oligonucleotide, and then DNA polymerase can come fill in the gap and ligase seals it shut, which is kind of like what we saw in the DNA replication slide. If the enzymes involved in this repair pathway are mutant or deficient, this causes a disease called xeroderma pigmentosum. This is a really serious disease, and people who have it rarely live past the age of 20. Cells in these patients can't repair pyrimidine dimers, so they're particularly sensitive to sunlight and need to avoid it completely if possible, and they usually die of melanoma or squamous cell carcinoma. Here's a picture of squamous cell carcinoma in one patient. Base excision repair is a second type of repair mechanism which fixes bases that have been covalently modified, and it starts just the way it sounds. The damaged base is excised after being recognized by glycosylases. So let's say you have a stretch of DNA like this, and maybe this cytosine gets deaminated. To repair it, the glycosylase that's specific for that base, which is cytosine in this case, will come over and remove just the base. This results in an apurinic or apyrimidinic site, or AP site, and either just that single nucleotide is removed, which means that the purine or pyrimidine base has been removed. Next, an AP endonuclease cuts the DNA next to the AP site, and either just that single nucleotide is removed, or a string of up to 10 nucleotides are removed. Once again, DNA polymerase fills in the gap and ligase seals it shut. The last type of single-strand repair pathway is mismatch repair, and this fixes mutations that have resulted in a DNA mismatch, which is when bases that are lined up in a complementary sequence don't match. For example, in this case, the A and the C don't match. This can happen during DNA replication, since the DNA polymerase proofreading is good but not perfect. Now, to repair this is a little more complicated than the two pathways we've talked about already, but in both of those it was easy to tell where the damage was. Thymidine dimers and covalently modified bases are obviously not normal, but in this case, it's not immediately clear which strand is normal and which is mutant. Should this A be bound to a T, or should the C be bound to a G? To handle this issue, cells can look at methylation. Some cytosines are methylated for a few reasons, including regulation of gene expression in CpG islands, and the strand that is not methylated must be the new strand, since it takes some time after DNA replicates for methylation to happen. So this is probably the one with the error, and it must be repaired. From this point, it becomes pretty similar to what we already discussed. Endonucleases will come nick the mutant strand upstream and downstream from the mutant base, then exonucleases remove that piece of DNA, and DNA polymerase comes and fills it in, and ligase seals the holes. If enzymes involved in mismatch repair are mutant or deficient, patients have a high prevalence of hereditary non-polyposis colorectal cancer, or HNPCC. This isn't nearly as bad as xeroderma pigmentosum that you get with a defect in nucleotide excision repair. Patients with defects in mismatch repair typically don't get cancer until they're in their 40s, and some will never have any serious problems. That should give you an idea of how accurate DNA synthesis usually is, since the wrong base is added so infrequently that even if you can't fix it, you might never have any problems. The last repair pathway I'll go over fixes double-strand breaks. So let's say we have this double-stranded DNA, and it gets broken apart. Well, we need to put them back together again, and this is called non-homologous end joining, because it joins together the two ends of double-stranded DNA, which are not homologous, since they are clean breaks with no overhanging end. The mechanism of this one is low yield for your exam, so I won't go through it, but the clinical consequences are important. First of all, what causes double-strand breaks? It's most commonly X-ray radiation. So patients that have a defect in one of the enzymes involved in this repair pathway would be particularly sensitive to X-rays and CT scans, and you should avoid these if possible. Also, the VDJ recombination pathway, which is used by lymphocytes to make random immunoglobulins and T-cell receptors, uses non-homologous end joining. So patients who are deficient in this pathway can have severe combined immunodeficiency. I also want to mention ataxia telangiectasia. So ataxia telangiectasia is caused by a defect in the gene ATM, which stands for ataxia telangiectasia mutated. And guess what this gene does? It's involved in non-homologous recombination. So if ATM is mutated, you get this pretty severe disease that includes ataxia, which is basically poor coordination, telangiectasia, which are dilated blood vessels near the surface of the skin, and immunodeficiency, since you can't do VDJ recombination. DNA, RNA, and protein synthesis direction is not particularly high yield, but I'll go through the important parts. You should definitely know that both DNA and RNA synthesis proceed in the 5' to 3' direction, and proteins are synthesized from the N to the C terminus. It might help you to think that 5 to 3 rhymes with N to C. So remember, 5 to 3, N to C. Know what the 5' and 3' even mean? These numbers correspond to the carbons in the sugar, with the 1 being closest to the base, going all the way around to the 5 being next to the phosphates. Aside from that, you should know that the nucleotides are added to a growing strand by having the 3' hydroxyl group in the last nucleotide in the chain bind to the first phosphorus in the nucleotide triphosphate, which is attached to the 5' carbon. This leaves you with 1 phosphate group connecting each pair of nucleotides. This is important because some drugs that block DNA synthesis are basically just nucleotides that have modifications to this 3' hydroxyl group. So they're still able to be added to the DNA strand, but don't allow it to grow further. For example, the thymidine analog zidovudine, also called AZT, is used to treat HIV because the viral reverse transcriptase will add it to the growing strand, but then can't build off it any further, since it's missing the 3' hydroxyl group. This azido group helps it cross the lipid bilayer to get into cells. Stavudine is also used to treat HIV using the same mechanism, only instead of replacing the hydroxyl with an azido group, it's just been removed. Okay, on to our next flash quiz slide. What type of DNA repair fixes damage caused by UV light? So this one would be nucleotide excision repair, and remember, someone with a deficiency in this repair pathway would have xeroderma pigmentosum, and would develop skin cancer really easily when exposed to sunlight. Okay, you're probably getting sick of DNA by now, so let's move on to RNA. mRNA is basically used to send a message from DNA to the ribosome so a protein can be made. This figure shows a typical layout of an mRNA molecule. We'll come back to this later, but for now I want to point out that the coding region, shown in orange, is where the codons are, but there's actually a lot more to an mRNA molecule than codons. There's a specific codon that designates the start of the coding region. Do you know what that is? That's AUG, which codes for methionine. In prokaryotes, this codes for formylmethionine. In very rare occasions, it can also be GUG. The stop codons, UAA, UAG, and UGA, don't code for any amino acid, but instead are recognized by proteins called release factors and result in the termination of translation in the ribosome. Okay, let's take a step back and see how this messenger RNA came from DNA. Keep in mind that the translation start site, which is the AUG codon, is not the same place as the transcription start site, which is the beginning of the 5' untranslated region. And the same is true at the other end. There's a lot of stuff that's transcribed into mRNA after the stop codon, which is called the 3' untranslated region. Genes have a few parts to them at the DNA level. At the 5' end, there are promoters, enhancers, and silencers. These are just DNA sequences, not other types of molecules. Proteins like transcription factors can recognize these sequences and bind to them to activate transcription in the case of promoters and enhancers, or repress transcription when they bind to silencers. Generally, promoters are within a few hundred base pairs of the transcription start site, whereas enhancers and silencers can be tens of thousands of base pairs upstream or even within the introns. One of the most important parts of the promoter is called the tau box, which is an area about 25 nucleotides upstream from the transcription start site in eukaryotes and is enriched for T's and A's. A protein called the tau binding protein binds here and recruits other transcription factors. There's also a sequence called the cat box about 70 base pairs upstream from the transcription start site, and this helps regulate transcription too. The transcribed region here is made up of introns and exons. The exons are the parts that will actually make it into the final mRNA, and the introns will be removed by a process called splicing. Once again, the transcription start site here is not where the start codon is. If we compare this image to the one above, transcription begins here, and then we have the 5' UTR, and the start codon is a little further downstream. The main function of this 5' untranslated region is to regulate ribosome binding to mRNA. The 5' cap and 3' poly-A tail are added later, and were not part of the original DNA sequence. Also, make sure you keep in mind the difference between the template strand and the coding strand. The template strand will be complementary to the mRNA, and the coding strand will have almost the same sequence as the mRNA with two exceptions. Do you know what they are? The mRNA is only different in that it has uracil instead of thymine and has its introns spliced out. And to review from earlier, if mutations occur in promoters, it can change the amount of transcription of that gene, usually reducing it. I already talked about enhancers, silencers, and promoters, but I want to point out one more thing about how they work. Promoters, which are the region 1 or 2,000 base pairs before the transcription start site, are relatively straightforward. Proteins like the tau binding protein, or TBP, can recognize and bind them, and recruit RNA polymerase and other proteins to help out with transcription. But enhancers are thousands of base pairs away. So how do they work? Well, the way I've drawn this is a big clue, but they serve as docking sites for other proteins, and ultimately the whole DNA strand can bend around, so they can help regulate transcription too. There isn't a whole lot you need to know about RNA polymerases, but here are the key points. The easiest way to keep these straight is that they're numbered in the same order that they're used in protein synthesis from the perspective of the ribosome. Type 1 makes rRNA, which is the first RNA in the ribosome. Type 2 makes mRNA, which gets to the ribosome next, and Type 3 makes tRNA, which comes to the ribosome last. Type 2 is what opens DNA at the start of transcription, and none of them have any proofreading ability. Also, prokaryotes only have one type of RNA polymerase, which does everything. I also want to mention this toxin called alpha-amanitin, which is found in deathcap mushrooms. It inhibits RNA polymerase type 2, which probably affects most organs and systems, but clinically presents as hepatotoxicity. Alright, for our next flash quiz slide, can you tell me what amino acid is encoded by the codon AUG? This would be methionine, which is the start codon, although this can be used as a normal codon as well, not just at the start. So when RNA polymerase II first transcribes mRNA, it doesn't look like this nice colorful one here. It's first called heterogeneous nuclear RNA, or hnRNA, and needs to go through a few more processing steps in the nucleus before it's officially an mRNA. First, a 7-methylguanosine cap is added to the 5' end, which is literally just a guanosine molecule with an extra methyl group on it, but connected backwards. So its 5' end is connected to the 5' end of the mRNA by 3 phosphates, as shown here. Next, the sequence AAU-AAA at the end of the 3' UTR is used as a signal to add on about 200 more A's to the end, which is called polyadenylation. Both the cap and the polyadenylation regulate nuclear export, prevent degradation by exonucleases, and promote translation. Third, introns have to be spliced out, which I'll go through in more depth in the next slide. But before we go on, can you think of any examples of genes or types of genes that do not receive 5' caps? Since the 5' cap is only added in the nucleus, genes in the mitochondrial genome would never be capped. Okay, let's talk about splicing. This is how introns are removed from hnRNA. We have this nice diagram in the book, so I'll use that. In this diagram, the intron is the DNA between exon 1 and exon 2, and this includes a few key nucleotides. First, the transcript combines with the small nuclear ribonucleoproteins, sometimes called SNRPs, to form a structure called the spliceosome. The SNRPs recognize specific sequences in the transcript and cause the DNA at the beginning of the intron to detach from that exon as this phosphate binds to a specific adenine within the intron, forming a loop called the lariat. Here you can see what it looks like once the loop is formed. Exon 1 is then joined to exon 2, and the lariat is no longer attached to anything, so it floats away and is degraded. This lariat contains the intron, so after doing this to all the introns, you end up with only exons. The concepts here are more important than the details for your exam, but one detail that could be tested is that there's a certain disease in which patients make antibodies to snRNP proteins. Any idea what this is? This would be lupus. We've already covered a lot about introns and exons, but one last thing I want to cover here is the concept of alternative splicing. So you've got all these exons, but you don't need to use all of them when you splice them together. Exons can be combined in different combinations to create different versions of the protein, and exons often correspond to specific protein domains. For example, maybe there's a protein that has a DNA binding domain, a catalytic domain, and 2 regulatory domains, so it can be repressed by other proteins. If for some reason a cell only wanted one of these regulatory domains to be present in certain situations, it can do that by keeping or removing that exon in the mRNA. This saves a lot of space in the genome compared to having 2 entire separate genes, which is especially useful for viruses since they have a very limited space for their genome. Alternative splicing is another area in which mutations can cause disease, though, with a key example being beta thalassemia. In beta thalassemia, a gene that codes for the beta-globin protein in hemoglobin has a mutation in part of the gene that normally allows the spliceosome to recognize the intron-exon boundary. The mutation prevents proper splicing, so you end up with an incorrectly spliced mRNA which codes for semi-functional or dysfunctional protein. Alright, for our next flash quiz, let's revisit mRNA modifications. So, is the 5' cap added to the mRNA before or after it leaves the nucleus? So this would be before it leaves the nucleus, because remember, the 5' cap, just like the poly A tail, is necessary for export from the nucleus.